Welcome everyone. Today we will discuss about cytoskeleton and molecular motors. So let's begin with the discussion. First of all, what is cytoskeleton? So these cytoskeletons are present in the eukaryotic cells and they have an internal scaffold protein, the cytoskeleton. And these cytoskeleton control the shape and movement of the cells as well as the organelles present within the cell. Okay, the cytoskeleton consists of number one microfilament, number two intermediate filaments, and number three microtubules. Okay, microfilament, intermediate filaments, and microtubules. We will discuss about all these three in detail, starting with number one microfilaments. So these microfilaments are very small in size. They have the diameter of around 5 to 9 nanometer and they are helical polymers of the protein actin. Okay, they are formed by the polymerization of the protein actin, and these actin have the role in mechanical support of the cell. Next is my intermediate filaments. These intermediate filaments are slightly bigger than the microfilaments. They are about 7 to 11 nanometer in diameter and they are rope-like structures. Okay, and they also provide the mechanical strength and the resistance to shear stress. The third type is microtubule. They are even bigger than intermediate filaments and they are made up of protein tubulin. Okay, now these microtubule, the walls of the microtubule, they are made up of array of alternating alpha and beta subunits. Okay, so the mitotic spindle, if you remember the cell division, the mitotic spindle which is involved in separating the two chromosomes. Okay, the mitotic spindle which is involved in separating the two chromosomes, these spindles are made up of what? These spindles are made up of microtubules. Okay, this thread like structure which is involved in separating the chromosome, they are made up of microtubules. Okay, there are drugs like colchicine, vinblastine, they inhibit microtubule formation, whereas another drug which is taxol, it stabilizes the microtubule. Okay, so these are very common and they are often asked. Okay, so these since these drugs interfere with the mitosis, these compounds are sometimes used as an anti-cancer drug to stop the division of the cancerous cells. Next is molecular motors. These motor proteins, they bind to cytoskeletal filaments. Okay, cytoskeletal filaments means what? Microfilament, intermediate filament, and microtubules. Okay, so these molecular motor proteins, or motor proteins, they bind to cytoskeletal filament and they use the energy derived from ATP hydrolysis. Okay, these molecular motor proteins, they have the head region and the tail region. Now, this head region are responsible for hydrolyzing the ATP. Okay, and the tail region binds to the cargo protein. Okay, so this molecular motor, they have the head region and they have the tail region. The head region is responsible for ATP hydrolysis and the tail region binds to cargo. Now, this is again an important topic from where the question comes. So what are the examples of the motor protein? They are myosin, kinesin and dynein. Okay. Next is muscle structure. So within every vertebrate, the muscle contains within the sarcoplasm. So within the sarcoplasm of the muscle, what is present? Many myofibrils are present. Okay, these myofibrils are made up of sarcomere units. Okay, so within the muscle, sarcoplasm is there. inside the sarcoplasm, or sarcoplasm is made up of myofibrils, and these myofibrils are made up of repeating sarcomere units. Okay, so what is the structure of each sarcomere unit? So within the sarcomere are the alternating dark A band and light I band. Okay, so within the sarcomere, what we will see the dark band and the light band. Dark band is the A band and the light band is the I band. Okay, now within the dark A band, between the dark A band, there is the H zone. There is the H zone. Okay, within the dark A band or between the dark A band, there is a zone which is the H zone. Okay, and within or between the light band, there is a line which is known as Z line. Okay, within the light I band or between the light I band, there is a line known as Z line. Okay, so what I discussed, I said that muscle fiber are made up of sarcoplasm. Sarcoplasm contains many myofibrils, and these myofibrils are made up of repeating sarcomere unit, and this sarcomere unit consists of dark A band and light I band. The dark A band has a line in between which is the H line, and the light I band has a line in between which is the Z line. Now another thing, this myofibril, it consists of two types of filament, thick filament and thin filament. Okay, thick filament and thin filament. The thick filament are made up of myosin, and the thin filament is made up of actin. Okay, actin, tropomyosin, and troponin. We will discuss about all that in detail. Okay, so when a muscle contract, very important, please focus, when a muscle contract, these thick and thin filament which is myosin and actin, they slide over one another so that there is the sarcomere length is shortened. Okay, so when a muscle contract, thick and thin filaments slide over one another, and it causes a shortening of the length of the sarcomere. So what is sarcomere? It is a repeating unit of myofibril. Okay, and it consists of dark band and light band. Clear? Now coming to myosin, which is the thick filament. Now the protein myosin consists of two heavy polypeptide chains and two light chains, two heavy and two light chains, and these are arranged as a double-headed globular region attached to a two-stranded alpha helical coiled coil. So I will show you the structure. It would look somewhat like this, with a double-headed globular region which is attached to a two-stranded alpha helical coiled coil. Okay, so this will look somewhat like this. This is alpha helical coiled coil, and this is double headed globular region. Okay, so these myosin molecules, they assemble into filaments. They are able to hydrolyze ATP and they are able to bind actin. Okay, they have binding site in their head region and they can hydrolyze ATP. Now coming to the next, actin. Now actin, they are the constituent of the thin filament. Now we are coming to thin filament. Okay. So this thin filament, they can exist in the monomeric form or the polymerized form. So the monomeric form is known as G-actin and the polymerized form is known as F-actin. And these actins are connected to thick filament. Thick filament means what? Myosin. So these actins are connected to myosin by a cross bridges which is formed by S1 head of myosin. So in the head region of myosin, there is the region where actin can bind. So this region is S1 region of myosin, which binds with the actin forming cross bridge. Next is the generation of the force in the muscle, how the force is generated in the muscle. So there is a cyclic formation and dissociation of the complexes between the actin filaments and S1 head of myosin. So I told you that S1 head of myosin can interact with the actin filament. So this formation and dissociation of these complexes, which complex? The actin filament and S1 head of myosin. When these complexes form and dissociate, it leads to the contraction of the muscle. So when actin binds with myosin, what happens? Myosin releases its ADP and PI. Okay, when actin binds with myosin, then myosin releases its bound ADP and PI. When this ADP and PI is released, what happens? There is a conformational change. What happens after this change? After the conformational change, what is seen? That the actin filament, they slide over the myosin filament. Okay, so the actin filament, they it will move over the myosin. Okay, once this process is done, then ATP will bind to myosin. So after this sliding process happens, the ATP will bind to myosin. When ATP bind to myosin, then actin is displaced. When ATP binds to myosin, actin is displaced, as a result of which the initial or the original stage is returned. So once again I repeat, actin when binds with myosin, myosin releases its bound ADP and PI. As a result of which there is a conformational change, and actin filament then slide over the myosin filament, after which ATP will bind to myosin. When ATP will bind to myosin, actin will be displaced from myosin and the S1 head returns to its original conformation. Okay, next we have to study about troponin and tropomyosin. So what they are? So I already told that they are along the thin filament, and this tropomyosin, very important, please note down, this tropomyosin have the role in preventing the association of actin and myosin. When, in what condition will it prevent? In the resting stage. Okay, so in the resting stage, tropomyosin will prevent the association of myosin and actin. Okay, it will prevent the association of actin and myosin in the resting stage. Now, another important point, that calcium ion, these calcium ion are released from where? From the sarcoplasmic reticulum. So from sarcoplasmic reticulum, the calcium ions will be released into sarcoplasm. Once calcium ions into the sarcoplasm, so when, under what condition will calcium ion will be released? It will be released in response to a nerve stimulation. Okay, so these calcium ions, they bind to the TNC subunit of troponin. Okay, so calcium ion, they binds to the TNC subunit of troponin. After which what happens? That the troponin conformation will be changed, and this conformational change, this is transmitted. Okay, this movement is transmitted by an allosteric mechanism through TN1, TNI and TNT subunits of troponin to tropomyosin. Okay, so once The conformational change occurs in T and C subunit of troponin, then through T and I and T and T subunit of troponin. What happens? Is this mechanism is transmitted through tropomyosin. So when, due to the stimulation of calcium, when this is transmitted to tropomyosin, then this tropomyosin will be inhibited, or the prevention by tropomyosin will be removed. So tropomyosin will move out of the way and will allow the actin and myosin to reassociate. So it is saying that in resting stage there will be no association between actin and myosin due to tropomyosin. Okay, but when there is nerve stimulation, whenever there is nerve stimulation, calcium ions will be released from sarcoplasmic reticulum into sarcoplasm, as a result of which the TNC subunit of the troponin will be stimulated, and this causes a conformational change in this troponin. This movement will be transmitted by an allosteric mechanism to another subunits of troponin, which is TNI and TNT, and ultimately transmitted to tropomyosin. So when tropomyosin receives this signal, it will move out of the way and will allow again the actin myosin to reassociate. Is that clear, guys? Another term is cilia. So these cilia are hair-like protrusions. They are present on the surface of the cell and they consist of microtubules. Very important to remember, very important to know also, because these are also from where questions are asked. So they are also made up of microtubules, and the microtubules in a cilium, they are bundled together in a characteristic 9 plus 2 arrangement. I will show you the picture in the next video. This is just an overview of the cytoskeleton chapter. So they are arranged, I'm just drawing a rough picture. They are arranged in 9 plus 2 arrangement within the axoneme. The outer 9 microtubule doublets look like a figure 8 with a smaller circle. So you can see here that the outer 9 tubules look like a structure of 8 with a circle and with subfiber A and subfiber B. This subfiber A circle is smaller and subfiber B circle is bigger. Okay. Next is dynein. So these dyneins are very large proteins and they form cross bridges with B subfibers. Okay, they are B subfibers are the large subfibers. So they form cross bridges with B subfibers and they have ATPase activity. Now, two dynein arms, they protrude from subfiber A, which upon hydrolysis of ATP move along adjacent B subfibers. Okay, I'll show you the picture in the next video where the two dynein arms from subfiber A, they hydrolyze the ATP. After ATP hydrolysis, these arms move along the adjacent B subfibers. Now, very important also from the questions have come in many exams, due to extensible Nexin links between the doublets, this sliding motion is converted into local bending of the cilium. Okay, so Nexin is the protein which helps in cilium bending. Okay, this is also an important point. So this is where I will end my video. So what we discussed in this topic is that So let's have a quick summary that we discussed about cytoskeleton, that these cytoskeletons, they control the shape and movement of the cell. The three types of cytoskeleton like microfilament, intermediate filament and microtubules. Microfilaments, they are very smallest. They are smallest among the three cytoskeletons, and they are formed of polymers of actin, and they give mechanical support to the cell. Next is intermediate filaments. They are around 7 to 11 nanometer diameter, and they are rope-like fibers, and they give mechanical strength and resistance to shear stress. Okay, the third is microtubules, which is made up of protein tubulin. They are bigger than intermediate filaments, and they consist of alternating alpha and beta subunits. Okay, so this mitotic spindle, which is involved in separating the chromosomes, they are made up of these microtubules only. So the drug like colchicine and vinblastine, they inhibit microtubule formation, but Taxol is a drug which stabilizes the microtubule, So since they interfere with mitosis, they are sometimes used as anti-cancer drugs. Next is molecular motors. So molecular motors, they bind to cytoskeletons and they hydrolyze the ATP to get the energy. Now these motor proteins have the head region and the tail region. The head region are responsible for ATP hydrolysis, and the tail region binds to the cargo protein. So three types of motor proteins we need to discuss: myosin, kinesin and dyneins. Now muscle structure, these muscles contains, vertebrate muscle contains sarcoplasm, and these sarcoplasm have many myofibrils, and these myofibrils are made up of repeating sarcomere units. Okay, these sarcomere units have alternating dark A band and light I band. Okay, within the dark A band there is a H line, and within the light I band there is a Z line. So whenever the muscle contracts, okay, one more thing, these myofibrils contain the thick and thin filament. The thick filament is myosin and the thin filament is actin, tropomyosin, troponin. Okay, so whenever a muscle contracts, the thick and thin filament, they slide over one another, causing the shortening of the sarcomere. Next is myosin. So we are studying the structure of a muscle. Next is myosin. These myosin consist of two heavy and two light chain. They have a globular head region and the alpha helical coil region. Okay, the head region are responsible for ATP hydrolysis, as well as the head region can bind to actin. There is a cross bridge formation between the actin and myosin. The actin, when it is in monomeric form, it is known as G actin, and when it is polymerized, it is F actin. Okay, the actin are the part of thin filament. Next is the generation of force in muscle. So how the force is generated? I told you that there is a cyclic formation and dissociation of the actin filament and myosin. So which area of the head region of myosin binds actin? So S1 head region of myosin binds to actin. And this actin and myosin interaction is constantly association and dissociation. Okay, this association and dissociation leads to contraction of the muscle. Okay, so whenever actin binds to myosin, it leads to the dissociation, or it leads to the release of bound ADP and PI of myosin. So bound ADP and PI is released from myosin when actin binds to myosin. Okay, when ADP and PI is released, there is a conformational change, and as a result of which actin slides over the myosin, and then ATP binds to myosin. Once ATP is bind to myosin, actin is displaced and the original conformation of S1 head is restored. Next is troponin and tropomyosin. So in the resting stage, when there is no stimulation, this tropomyosin prevents the association of actin and myosin. Whenever there is nerve stimulation, in that case, C plus calcium ions are released from sarcoplasmic reticulum into the sarcoplasm. Okay. So upon this stimulation, the T and C subunit of troponin, it okay, the this So these calcium ions, they bind to T and C subunit of troponin and cause a conformational change in this protein, as a result of which this movement is transmitted by an allosteric mechanism to two another subunits of troponin, which is TNI and TNT. So this TNI and TNT will transmit the signal to tropomyosin, and then tropomyosin will move out of the way, that I am not needed right now. There is an association between the anactin and myosin is required. Okay, next is cilia. The cilia are hair-like protrusions and they are made up of microtubules. So microtubules in the cilium, they are arranged in 9+2 manner, and this outer 9 microtubules, they look like a structure of 8 which have the central circle and the subfiber A and subfiber B. Okay, this is subfiber A, this is subfiber B. The subfiber A is smaller and the subfiber B is larger. The last is dynein. So these dynein are very large proteins and they form a cross bridge with B subfibers. Okay, they form a cross bridge with B subfibers. Upon ATP hydrolysis, they move along the adjacent. Okay, I'm sorry. So these dyneins, they protrude from subfiber A, but upon ATP hydrolysis, they move along the adjacent B subfiber. And due to the presence of nexin, the cilia can bend. So that's it for the overview of cytoskeleton. I will discuss in a little more detail in the next video. Thank you.